Approximately 38 million people in the world are currently living with human immunodeficiency virus, otherwise known as HIV, classifying HIV as a global epidemic. Although the immune consequences of this virus are heavily studied, the neurocognitive effects in the central nervous system are much less well known. Combination antiretroviral therapy, or CART, has been shown to be effective in reducing HIV-related deaths, but has been ineffective for long-term treatment. Even with this treatment, viral protein reservoirs have been found in HIV patients, and one such protein is the transactivator of transcription, or TAT, which drives disease progression. TAT has been shown to cause excitotoxic effects and induce inflammatory responses which can damage neuron structure, leading to region-specific deficits. Motivational deficits in HIV-1 patients are associated with this TAT-induced neuron injury, specifically in the striatum. It's been shown that the upregulation of the endocannabinoid 2-AG serves neuroprotective function by reducing neuroinflammation and excitotoxicity associated with TAT. MJN110 is an effective monoacylglycerol lipase inhibitor, which limits the degradation of 2-AG and has been shown to similarly serve this function against TAT-mediated excitotoxicity. In a previous study conducted in the Fitting Lab, investigating the effects of TAT and MJ110 on behavioral flexibility, it was found that there was a potential motivational factor present in TAT-positive subjects that was then later altered by MJ110 administration, which prompted this current study. So my current study employs an operant conditioning task to study the effects of HIV-1 TAT and MJ110 on motivated behavior. 36 female doxycycline-inducible TAS transgenic mice were separated into 4 groups based on genotype and treatment. Half of each genotype was treated with MJ110, the other half of each genotype was treated with a saline vehicle solution. In this task, mice were trained to associate a no-spoke response with a sucrose reward on a fixed ratio 1 schedule, and then advance to a single progressive fixed ratio test upon meeting criteria. Breakpoints, among other measures for motivated behavior, were the primary proxies for motivation. Immunohistochemistry was then performed to visualize neuron structural integrity and cellular inflammatory response. So we found that there were no significant main effects of treatment or genotype or interaction between these factors for any of the motivated behavior measures. However, in the IHC data, we did find that MJ110 acts significantly more restorative towards neuron structural integrity in TAT-positive subjects, but more damaging in TAT-negative subjects. Since these contrasting trends between genotype align with the previous study from the Fitting Lab, this could be due to a result of immune tolerance because of long-term TAT exposure, an overactive endocannabinoid system, or differential pro-inflammatory protein expression between genotypes. Further studies assessing involved brain regions, other endocannabinoids, and pro-inflammatory protein expression should be conducted to better elucidate our behavioral responses and variation due to TAT.